Hello everyone, welcome back to the new session in biochemistry, and here we will be continuing with the previous presentation, that is a glycolytic pathway. So glycolysis, as already defined, it is the sequence of reactions that will break down a single molecule of glucose to two molecules of pyruvic acid along with the production of ATP molecules, right? And the glycolysis can be differentiated in two steps. The first one being the preparatory phase or the priming phase, or it is otherwise known as activation or energizing phase, where the glucose is getting activated or energized to produce glyceraldehyde 3 phosphate. Okay, so here what happens is glucose is getting phosphorylated and it is converted into a 3 carbon phosphorylated sugar. Okay, glucose is a 6 carbon sugar, so 6 carbon glucose, it is getting converted into 3 carbon phosphorylated sugar. Okay, and here it is an energy investment phase where ATP, it is getting utilized at various steps for getting phosphorylation process happen. Now the second phase, it is a payoff phase, that is energy generation phase. It is actually where the glyceraldehyde 3 phosphate, which is molecules, two molecules of glyceraldehyde 3 phosphates are produced, and these molecules are getting converted into pyruvic acid through various steps. And during this this particular phase, ATP is generated, and hence it is also referred as energy generation phase. Okay, so glycolysis gets completed in two phases: preparatory phase, or it is otherwise known as priming phase. Priming means glucose is getting what you call prepared for, or it is getting activated, or it is getting phosphorylated for getting into the glycolytic pathway. Okay, so it is preparatory phase, and this involves conversion of 6 carbon glucose to the 3 carbon glyceraldehyde phosphate. Okay, then here ATP, it is getting utilized at two steps, right? A single molecule of glucose is getting converted to two molecules of glyceraldehyde 3 phosphate. Now in the next phase, that is a payoff phase, that is energy generation phase, where uh, a, each of the molecules of what you call glyceraldehyde 3 phosphate, it gets converted into 3 carbon pyruvic acid, and during this ATP molecules are produced at various stages. ATP molecules are produced. Okay, now so we will see in detail what happens in each of the phases. The first one is the preparatory phase. Preparatory phase, as already mentioned, it involves phosphorylation of glucose and its conversion to glyceraldehyde 3 phosphate. Okay, glucose molecules, how it reaches the cell. So after intake of food, after its digestion in the alimentary canal, after its absorption at the intestine, uh, the blood glucose, the glucose enters the blood, and the blood glucose level increases. The blood takes the glucose to various tissues and cells in need of the glucose molecule, and there the glucose will enter the cell from the blood uh, by actively or passively, whatever it is, because it is glucose is permeable to the cell membrane, and maybe in some cases it may also require the help of certain transporters, which we call as GLUT or glucose transporter molecules. In whichever way it is, glucose will enter the cell. Now glucose, since it may be permeable to the cell membrane, it may come get transported back. To avoid such conditions, glucose have to be fixed inside the cell, and this is done with the help of phosphorylation process, or we call it as a first priming reaction. Okay, so glucose gets phosphorylated to produce the glucose 6 phosphate. Glucose 6 phosphate, it is impermeable to the cell membrane, and we can see that this glucose 6 phosphate, it becomes a substrate in various metabolic reactions, for example like glycogenesis, gluconeogenesis, etc., and it is a very important molecule. Okay, coming back to the process, the glucose, it gets phosphorylated to glucose 6 phosphate with the utilization of ATP molecule. So the terminal phosphoryl group of ATP is getting transferred to glucose for the production of glucose 6 phosphate. Any enzyme which catalyzes the transfer of a terminal phosphoryl group of ATP onto a substrate, we call them as kinases. Okay, and here specifically we can see it is a hexose sugar onto which the terminal phosphoryl group is getting transferred to, and hence the enzyme is known as hexokinases. Okay, now hexokinase, if we see, there are four types of hexokinases: hexokinase 1, 2, 3, and 4, given Roman numbers 1, 2, 3, and 4. The hexokinases 1, 2, and 3, they are found in various cell types, and they can phosphorylate like glucose, mannose, fructose, etc. Okay, to its phosphorylated forms. Um, Now, the hexokinase 4, they are found on the hepatocytes or the liver cells and the beta cells of the pancreas. And these are responsible for the type 4 hexokinase. It is specifically responsible for phosphorylation of alpha D glucose. And hence, type 4 hexokinase is also known as glucokinase. And these four different types of hexokinases are being called as isozymes. I hope you remember the isozymes which we discussed in under enzymes. The isozymes of these hexokinases, they actually catalyze similar reactions in various cells, but what happens is structurally maybe they may be different. That is, chemical composition of the enzyme may be different. Then it is also one more thing regarding hexokinases is that they are gene products of different genes. They are products of different genes. Okay, so just because they take up similar reactions, they catalyze similar reactions, they have been named as hexokinases, right? It is 1, 2, 3, and 4 different types. These four types are referred as the isozymes of hexokinases. Now, coming back to the glycolytic pathway, the glucose, we can see that it is getting phosphorylated to form glucose 6-phosphate, and this reaction is irreversible process. That is, the same enzyme cannot bring about the reversible reaction. Uh, Getting the glucose back from glucose 6-phosphate cannot happen with the help of hexokinase. Okay, so it is a irreversible process. Another thing is, like, or just like many other kinases, we can see hexokinase require magnesium ion, the presence of magnesium ions as a cofactor. Okay, now the next reaction is the isomerization process. So the first priming reaction or first phosphorylation reaction, it is glucose to glucose 6-phosphate. And I hope you understood why the name has come hexokinases. The kinases, wherever phosphorylation happens with the help of ATP, the enzyme involved will be a kinase. Okay, now the next is isomerization process, where glucose is getting converted to fructose. I hope you remember glucose is a aldohexose and fructose is a ketohexose, right? So it is getting converted, and this is actually an isomerization process. And you can see glucose 6-phosphate, it get isomerized to the fructose 6-phosphate, and this particular process, it is a you can see reversible process, and the enzyme involved is phosphohexose isomerase. The isomerization process, and hence the enzyme is isomerase. And since the isomerization takes place between phosphorylated hexose sugars, the name enzyme has got the name phosphohexose isomerase. Okay, I hope you will remember how the name has come, the enzyme name. Okay, now next one is, you can see here, this is a fructose 6-phosphate, right? So glucose 6-phosphate, where the sixth carbon is having the phosphoryl group, while the in the fructose 6-phosphate, the sixth carbon is having the phosphoryl group over here. Right now, what happens next is again the second priming reaction or second phosphorylation reaction, where fructose 6-phosphate is getting phosphorylated to fructose 1,6-phosphate. Okay, this is bisphosphate. We'll see why the name has come. Now, here what happens is, apart from this phosphoryl group on the sixth carbon, the fructose 6-phosphate also gets phosphorylated on the first carbon. Right, so you have the first and the sixth carbon being phosphorylated. Now, since the two phosphates are on two different carbons, the name has come bisphosphate. If the two phosphoryl group were on the same carbon, one after the other, the name would have come diphosphate. I hope you remember adenine diphosphate, right? It was because the two phosphates were added on the same carbon, one after the other. But here it is two phosphates are added, but on two different carbons, and hence the name fructose 1,6-bisphosphate. Okay, so the enzyme is phosphofructokinase. Why the name kinase? Because it is phosphorylation with the help of ATP. Now, why the phosphofructo? Because it is phosphorylated fructose, it is getting phosphorylated again. So that is why the name phosphofructokinase. Okay, so the second priming reaction involves the phosphorylation of fructose 6-phosphate to fructose 1,6 bisphosphate, and this is a again, you can see it is an irreversible process, and this requires ATP, and the enzyme involved is phosphofructokinase 1. Fine. I hope that part is clear. Now, the next step we have is the conversion of fructose 1,6 bisphosphate. Again, this is also a 6 carbon sugar. Till here, they are 6 carbon sugars. Okay. Coming over here, what happens? This involves splitting phase, okay, or the step which involves the splitting of fructose 1,6 bisphosphate. Here, the cleavage of 6 carbon sugar phosphate to 3 carbon sugar phosphate. So, what has happened? The fructose 1,6 bisphosphates, it is being cleaved to produce 3 carbon sugars, phosphorylated sugars. Okay, and these are glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Right. So you can see here the what you call the fructose 1,6 bisphosphate. It is getting split or cleaved to produce two different triose phosphates, and they are glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Okay, and this particular reaction is again a reversible process, and it is being catalyzed by the enzyme aldolase. Okay, uh, you can see here this is aldose sugar, right, aldehyde containing sugar, and this is a ketose sugar. Again, this can be isomerized. These two can be isomerized, and that is dihydroxyacetone phosphate can get isomerized to produce glyceraldehyde 3 phosphate. Okay, so and this is done with the help of the triose phosphate isomerase. Okay, that is interconversion of the two triose phosphates happen. Only one of the two triose phosphates formed by the aldolase enzyme can be directly degraded in the coming steps, subsequent steps of glycolysis. That is, to only one of the triose phosphates will enter the payoff phase. Okay, so before that, what happens is these two get interconverted. Usually, glyceraldehyde 3 phosphate is the sugar which will enter the payoff phase. So, dihydroxyacetone phosphate, it will get isomerized to produce the glyceraldehyde 3 phosphate with the help of triose phosphate isomerase. Here it is isomerization process taking place. As already mentioned, these two are trioses, that is 3 carbon sugars. Okay, this is an aldolase, sorry, what do you call aldose, and this is a ketose. Okay, it is getting interconverted. So isomerization process, which will be catalyzed by isomerase enzyme. Okay, so what kind of sugars are getting isomerized here? It is a triose sugar, and it is phosphorylated. Hence the name triose phosphate isomerase. I hope that part is clear. Okay, so only glyceraldehyde 3 phosphate will enter the payoff phase. Okay, so all the sugars which are produced, different types of sugars which are produced from fructose 1,6 biphosphate through the cleaving, it will get converted into glyceraldehyde 3 phosphate. Right, so only glyceraldehyde 3 phosphate will enter the payoff phase. And in the payoff phase, what is happening? It is oxidative conversion of glyceraldehyde 3 phosphate to produce the pyruvic acid, and this is actually the whole set of reactions in payoff phase involves the production of ATP molecules at various stages. Okay, and the payoff phase will be described in the next presentation. Fine, thank you.